feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. And I'm right where I belong in my happy place. And I'm right where I belong in my happy place.
Welcome to the National University of Singapore Yong Lulin School of Medicine Healthy Longevity webinar. Thank you for joining us. Assistant Professor Yifan Yang is with us tonight talking about compression of morbidity, being healthier for longer, escaping age-related diseases. I would like to remind you, to use the Q&A function to send question, remarks, and other suggestions you have. We are starting with a short presentation, and this time today by Jessica Liu. She is our PhD student at the NUS Academy for Healthy Longevity, and she will present her PhD work entitled Digital Biomarkers of Aging for Monitoring Physiological Systems in Community-Dwelling Adults.
Thank you very much. As mentioned, my name is Jessica, and I'm very excited to share with you about digital biomarkers of aging for monitoring physiological systems in community-dwelling adults. So to start with, what are biomarkers of aging? Well, the Biomarkers of Aging Consortium, which comprises of researchers in the field from all over the world, they came up with a framework for the terminology and characterization of biomarkers of aging, including classification and potential clinical use cases. And they divided the biomarkers of aging into three categories: molecular, physiological, and digital. And they defined a biomarker of aging as a measurable sign in your body that tells us your biological age, so how old your body seems to be functioning regardless of your actual age. And it can be a single measurement or a combination of parameters, and ideally we would be able to see improvements in this biomarker if healthy changes are made. So the focus of this publication was on the molecular biomarkers of aging, which includes epigenomics, transcriptomics, proteomics, and metabolomics. However, the gap was that the landscape of digital biomarkers in aging and longevity has not been explored and mapped. So the digital biomarkers of aging have not yet been defined, and that's what our work sets out to do. We wanted to define the digital biomarkers of aging in collaboration with the Biomarkers of Aging Consortium, and in this work, we classified, characterized, and evaluated digital biomarkers of aging across 10 physiological systems, exploring their applications in research and clinical practice. So how do we start? We first started with a systematic search to identify minimally invasive or non-invasively measured digital biomarkers that would be suitable for longitudinal studies, so long-term use, as well as practical use by community-dwelling adults. And a biomarker was included if it can be measured using a digital device, which was defined as electronic devices equipped with sensors. So this can be wearable, portable, or suitable for use at home or during everyday activities. And it can be worn on the body or integrated into clothing. And portable devices just means that they could be implantables, ingestibles, any functions on your smartphones, as well as mobile apps. And next, we classified all of these digital biomarkers by their physiological system, 10 of which they're all defined by the American Physiological Society. And then after that, we characterized the capture method of each digital biomarker into dedicated wearable sensors, which includes accelerometers or optical heart rate sensors, dedicated or purposive technological solutions, which include games, interviews, or photographs, as well as non-dedicated technological solutions. So this would include human-computer interactions. While this list is not definitive or exhaustive, these are the possible devices or modalities that were used in digital biomarker studies that were found. Lastly, we evaluated the suitability of the digital biomarker as a biomarker of aging using criteria adapted based on the Biomarkers of Aging Consortium's framework. So this includes looking at its validity, whether it was age-associated, function-associated, and mortality-associated. We also looked at the generalizability of the digital biomarker, so can it be used in different populations, young or old, healthy or diseased, as well as the digital biomarker's responsiveness to intervention and its associations with clinical health outcomes. And lastly, it was also important for us to explore the cost for use in large-scale settings for these digital biomarkers. So overall, our work identified a list of digital biomarkers of aging for monitoring physiological systems in community-dwelling adults. And there were 8 physiological systems currently have measurable biomarkers of aging with available digital tools. And this work lays the foundation for how a digital biomarker of aging is defined and what is considered as a digital biomarker of aging. So this review has been accepted by the Lancet Healthy Longevity, and hopefully it will be published soon so everyone can read it. And if you're interested in participating in our clinical studies, do connect with us. Thank you.
Thank you, Jessica, and congratulations again for this wonderful work with a big consortium. So very well done to get everybody on board. It's a great pleasure now to introduce our speaker of tonight, Assistant Professor Yifan Yang, and he leads the Systems Biology Laboratory at Westlake University in Hangzhou, China, where he studies the complex dynamics of biological aging, statistical physics, and quantitative physiology. He holds a PhD from the University of Paris, very nice city, and completed his postdoctoral research at the Weizmann Institute of Science in Israel, working with renowned systems biologist, Professor Uri Allen. Yifan, it's a pleasure to have you here, really learning from you how we can compress even morbidity more. Over to you.
Thank you, Andrea. So tonight, I would like to share with you a project we recently finished with Professor Uri Allen at the Weizmann Institute of Science in Israel. I recently moved back to China to start my own group. So this is a very mathematical work project we did, and it's recently published on the journal Nature Communications. So the topic for this project is about compression of mobility. So in the field of aging research, we would like to, of course, extend lifespan, but consider a hypothetical scenario where the lifespan extended artificially by preventing death. But what you end up is a longer lifespan, but most of the extra years are spent in what we consider Sickspan. And of course, everybody considers this a nightmarish scenario. Alternative scenario that's sometimes pursued is what we call slower rate of aging. So in this scenario, the lifespan is extended, but the health span is also extended, but the proportion of health and sickness is held constant. In the 1980s, Dr. James Fries actually proposed a third scenario, where we actually postpone the onset of age-related morbidities and extend health span exclusively. And this is considered by a variety of authors to be the optimal trajectory of aging. Now, but the problem is that in both health research in humans and also longevity research in animals, very high quality longitudinal data are rare, and especially under longevity interventions. So instead of tackling this problem directly, we decided to look at another type of data that are perhaps more widely available. So this leads us to perhaps a figure that you've seen very often in publications, where there are two groups of animals. One is the control, shown in black here, and another group is the red treated group. And in this hypothetical experiment, the median lifespan is extended, so the author consider this a longevity intervention. And they usually publish their data in this format. However, there are actually more information embedded in the figure That's the shape of the survival curve. And that's what we're going to consider next. So in this slide, I'm showing you actually two experimental results of two separate interventions in mice. And the black, again, is the control group, the red is the treated group. And you can, on the left, you have the normal x-axis, where we show in real time, we show age in real time. So you see that both interventions are longevity interventions. However, in order to see the shape of the survival curve, what we do is we divide the age by the median age. So we will get a normalized age on the x-axis. And this way you can see the effect of the intervention on the shape of the survival curve. And in the top row is the intervention we consider to be scaling. So it's preserving the shape of the survival curve while extending lifespan. And the intervention on the bottom row is what we consider a steepening intervention. So as you can see, the renormalized survival curve now have a steeper curve. And in order to extract this information about the shape of the survival curves, we use a mathematical model to simulate the distribution of lifespan in the survival curve. So this is a recently published, discovered mathematical model where we're modeling the dynamics of a physiological damage called X. And we consider the fact that X is constantly being produced and removed. And also there's noise in this process. And this X drives the onset of morbidity and mortality. And this is basically a mathematical form of the geroscience hypothesis. And in two separate publications, we have shown that there's actually a qualitatively simple model that guides the fluctuation of damage in two different organisms. So in the first publication, in mice, the damage X is senescent cells. And in the second publication, where in a single cell bacterium E. coli, the damage is membrane disruption. And although the biological nature of damage in these two organisms are completely different, the circuit we find is actually strikingly similar, where age increases the production rate of damage, and at high damage levels, it inhibits its own removal, and thus leading to a vicious cycle. And we can simulate this model on the computer to get stochastic trajectories of damage. And you can see here that these are two simulated individuals, and they, because of noise in the system, they die at different times. And in order to model death, we apply a threshold called X critical. So now if we change the damage production rate in this model, we get what we consider scaling survival curves. On the other hand, if we change other parameters in the model, such as noise, damage removal, and mortality threshold, what we get is steepening interventions. So in fact, if we put a survival curve and we plot the steepness on the y-axis and longevity on the x-axis, we can use the location of a intervention to identify its mechanism of action by seeing whether it's affecting removal, which will result in the same steepness, or whether it's affecting removal, death threshold, or noise. In all three cases, they will result in longer longevity and also steeper survival curves. Now that we can link the shape of survival curves to damage dynamics, what we want to do is to use this to predict intervention's effects on health span and Sickspan. So first, I have to introduce the idea of how do we model health span and Sickspan. And this originated with an observation that many age-related diseases rises exponentially with age at the same rate as mortality. And this striking observation hint that perhaps there's a common mechanism leading to the onset of all these age-related diseases. So this leads us to modify our model by including a second threshold called XD here that's lower than mortality. And if the trajectory of an individual crosses the XT for the first time, we consider the onset of a disease. And just with this single additional parameter, now we can describe well the incidence rates of 90% of age-related diseases. However, there's also a few notable exceptions. One is osteoporosis in women, and the other is the incidence rates of Alzheimer's disease and dementia. Now, with this tool, we can simulate our models again for hundreds of thousands of individuals and calculate their onset of disease and death times. And we can calculate how much time they spend in disease versus in health. Now, again, if we affect the damage production rate, we'll get a scaling survival curve, meaning the shape of the mortality curve is the same, as you can see on the bottom left. And quite intuitively for us, this scaling intervention also results in approximate scaling of health span and Sickspan. So the relative Sickspan is held roughly constant. And this is intuitively understood because the production rate is the only place where age comes into our model. Affecting the production rate parameter eta is like stretching a rubber band. So you do get a longer lifespan, but you get the health span and Sickspan also prolonged proportionally. In contrast, if we affect, let's say, the damage removal rate by increasing it, what we get is a longer lifespan. But this time, as you see, we can get a steeper survival curve and also a compressed Sickspan. In fact, all three parameters that results in a steeper lifespan results in what we consider compression of Sickspan, as you can see in the bottom left with the red, the purple, and the yellow curves. The only exception where we have a steeper survival curve is exactly the artificial sustainment of life scenario I presented in the first slide, where all the parameters that relates to aging is not affected except the death threshold. And in this case, you get an expansion of Sickspan with a steepening survival curve. And intuitively, this is also relatively easy to understand once you look hard into the model. Because in our model, the only factor that carries noise in it is the damage. And it is also precisely this damage that leads to morbidity and mortality. So all three parameters that steepens the survival curve is in fact mitigating the effect of damage and thus delaying the onset of both mortality and morbidity. And now we see that the shape of the survival curve is linked with the relative Sickspan. In a mathematical model, we want to see whether this is true in real life. So we looked hard at model organisms, and indeed there are actually emerging evidence that this is the case. So we managed to find 4 separate studies, one in mice, one in flies, and 2 in worms, where they measure longitudinally the health span and Sickspan under various longevity interventions. And in all 4 cases, the relative Sickspan decreases with steepening survival curves. And this is as predicted from our model. And I'll walk you through one by one of these studies to give you more confidence that this is indeed the case. So in the first study by Luciano et al., they measured the health of mice under 4 dietary interventions, such as calorie restriction at 20%, 40%, intermittent fasting for 1 day versus 2 days. And they did this in 2 strains of wild-type mice just to have a proper control. And the way they measured health is what's called the frailty index of mice, where roughly 20 to 30 physiological parameters are measured, and the number of deficits are counted. And in the original study, they consider Sickspan to be any time of life where more than 3 deficits are counted. And in fact, the conclusion of our study is very robust to this threshold. So if we raise the threshold to 4 or 5, the negative correlation persists. The next study is a fly study where health is measured by local motor deficits, where these vials that contains the flies are banned against the counter to simulate a life-threatening challenge. So what the normal flies would do in this case is try to escape by jumping, flying, and climbing. And when these flies are really close to their death, they start to have locomotor deficit where they suffer problems in all kind of locomotor functions. And all these functions are carefully measured by this set of authors and summarized in this fantastic diagram. And then they shared this data with us. And they did this for 2 strains of Drosophila in both male and female flies under 6 dietary conditions. And under all these interventions, it's clear that the relative Sickspan decreased with steepness of survival curves. And the third organism we're going to talk about is C. elegans. So in C. elegans, it's relatively harder to measure health. There are generally 2 ways of doing it. One more widespread way is to measure the voluntary movement of worms. So when these worms get old, they stop moving, and people consider the time of not moving to be the onset of Sickspan. However, some authors argue that perhaps they're not moving because they don't want to. So instead, what they did on the right here by Stather and his co-authors is that they developed a device where an electric field is applied externally to these worms to stimulate them, to force them moving, and they measured the maximum power of their strengths, the maximum power of the muscles to define a Sickspan. And in both studies, we can see that the relative span decreases also with steepness. And in fact, we tested also different definition of steepness, and our conclusion is also robust to that. Now, we have tested our theory in real life. We now want to apply our theory to identify interventions that in mice that could perhaps compress the Sickspan by looking at the shape of their survival curves. So for this purpose, we used a fantastic resource, and some of you might know it, is the National Institute of Aging Long-Term Intervention Program. So it's a decade-long multi-site, open, and systematic investigation of mice longevity. So these are very well designed. They're based on a genetically heterogeneous stock of mice run in 3 different universities, and each university with a control cohort and a treated cohort with 50 female and 50 male mice each. And they're also open to new suggestions. So if you find a new drug that you think are going to prolong the lifetime of mice, you can suggest to them and they will test it for you. And lastly, they shared they publish their data that's documented with the life span of individual mice. And they also have their phenotype and genotypes, but the latter 2 cases are not these data are not widely not shared publicly. So these are their website where you see the interventions that are considered to be longevity interventions, and also the form where you can suggest new interventions. So we analyze the survival curves in this data set on 2 axes. One is the effect of the intervention on steepness, and the other is the intervention's effect on longevity. And you can see that most of the dots are around the origin. So this means that these interventions have relatively small effect size and doesn't reach the significance of statistical significance. However, The first classes of longevity intervention, we clearly see this in this data, are rapamycin-related interventions. In fact, this is the largest class of interventions because rapamycin is really well known to be a longevity intervention. So multiple dosage and multiple regimes are tested. And you can see almost all of these interventions lie on the right side of the graph. This means that rapamycin under all these dosage and regimes roughly extend longevity without steepening the survival curve. So rapamycin is indeed a scaling intervention. In contrast, there are about 4 types of interventions by mechanism of action that steepens the survival curve. So these are acarbose, which reduces the postprandial glucose spikes, BD, which I think simulates ketosis, and this steepens the survival curve only for males. The same for 17-alpha E2, which is a non-feminizing estradiol, and it also works only for males. And lastly is captopril, which is a ACE inhibitor that reduces hypertension. In addition to NIATP, we also look at the wider scientific literature, and we found 2 more classes of steepening interventions, and these interventions require genetic engineering of mice. And these are overexpression of VEGF, which also improves the vasculature, and also senolytics. And lastly, I also want to talk again about rapamycin, because rapamycin is the most robust longevity intervention. So in most of the data, it's a scaling intervention. But we do have one cohort that's at the bottom left, where the females mice are extended in a steepening fashion at high dosage. And in fact, this steepening effect can be seen as subthreshold. You can see the subthreshold steepening also in the lower dosage groups and in males, but only at old age. So our statistical tests don't pick them up. So we suspect that rapamycin indeed at old age has a secondary effect that's a scaling intervention. So finally, we have identified interventions that are steepening interventions in mice. We predict that these mice would prolong Sickspan. Now we want to shift our attention to another application of our theory when it comes to combination of interventions. So the idea is this. Given that we are able to use the shape of survival curve to pinpoint the mechanism of action of interventions, we can say, for example, rapamycin is a scaling intervention. So we suspect it operates through the damage production rate eta. In contrast, acarbose is a steepening intervention, so we think that it operates through the removal rate beta. So now if we combine these 2 interventions, because they have distinct mechanism of action, they're likely to be additive. And in this case, being additive is a very good news, because you get the longevity effect of both drugs, and you get the extra steepening intervention from the acarbose that will compress the Sickspan. So lucky for us, the NIATP tested this combination, rapamycin plus acarbose. And indeed, the data is consistent with our prediction. So the dashed eclipse are the theoretical predicted confidence interval, and the solid line eclipse is the actual data, the confidence interval for the data. And the 2 overlap very well with each other. However, this is only one drug combination. So we're looking for more drug combinations to test this idea. And lastly, we ask whether our theory can also be used to understand the steepness of human survival curves. So it's well known that the human survival curves has been steepening along with longevity. So what's shown here is the survival curves of Swedish birth cohorts born in the 1820s versus in the 1890s. And you can see after normalization, not only the median lifespan extended, the shape has also become more steep. So we were asked by reviewers why this is the case. So we apply our model to the human mortality data, but we notice that in human mortality data, the SR model alone doesn't produce a fit. And this is because at young age, there's there in the human mortality data, there are other causes of death that are age independent. So we call these age independent causes of death extrinsic hazard, and we just add a constant to the hazard predicted by the SR model, and this produced a fantastic fit to the to human data. And we apply this method to mortality data from 4 countries across the past century. And what we see is that there's no clear trend in the age-related parameter, such as the Gompers slope or the Gompers intercept. But the clearest trend in this data is the drop in extrinsic hazard. So what the dramatic improvement in life expectancy over the past 100 or 200 years are associated with preventing death from age-independent causes. And this highlights the need now, because the age-independent causes have dropped to almost zero. Now this is highlight the need to work on the biological mechanism of aging. And so this is, we hope that our method can be a contribution to that. So now I have to wrap up by first thanking the team that supported me in Israel during some tough times, including my mentor, Uriel Long, and all the model organism labs that shared the data with us, including Professor Konsulas from Greece, Professor Ham from Korea, and Professor Struthru from Spain, and all the various people that have provided us with funding and discussions. And lastly, I would like to pitch my own group in the beautiful city in Hangzhou, China, where we have started a new lab called System Aging in the Center of Interdisciplinary Studies at Westlake. So if you're interested in using computational methods to study aging and you want to live in the city of Hangzhou, you're very welcome. Thank you.
Thank you so much, Yifan. Brilliant presentation. I love the idea that we fixed everything except of the aging process itself. So hopefully that's really, I think we can do a little bit more, but I think let's fix aging. And that brings me to the first question. You really nicely showed that age-related diseases occur exponentially. Do you think that aging itself is also exponentially? And also maybe you can tackle if you think it's in mice or how humans would be different.
Right. Thanks, Andrea. So that's a very interesting question. I can tackle this perhaps in 2 ways. So the first answer is yes. So I do think that aging happens exponentially, not age as a number on the clock, but age as an actual biological age. So I do think that it happens very slowly and then quickly. And this onset of age, I think, is also more and more stochastic. So the onset of age-related is actually individual dependent. And so they happen exponentially and they vary among individuals. So that's one answer. And then I think we have some results using our model. So we have actually applied our model to various model organisms to identify how are these organisms different. And it turns out that, so I could perhaps share this briefly, is that mice is actually quite different from humans. So in our model, there's a critical age at which the repair system is completely saturated. So you have no way of fixing the damage at all. And this happens roughly for humans at age 110 and 115. But for mice, it happens actually at 18 months. So most of the mouse death actually happens after this critical age. So we think that the exponential process for mice probably happened earlier, and then there's something else going on for mice after age 18 months.
So do you think that the exponential one, that we can only measure it and that's exponential, or do you think that the origin of that damage we are having is also exponentially occurring?
So I think that probably most so we think that with aging dynamics there are several things going on. So one thing is that with a lot of physiological functions like basal metabolic rate or kidney filtration rate, these are well known to be linearly declining with age. But there are other classes of measurements, such as senescent cells and inflammation, and they accelerate with age. So we think that those happens exponentially. So it's a very interesting question, and I don't think we have a clear answer in terms of what differentiate between them. So we consider basically the exponential ones more to be the drivers of aging.
Yeah. I think if we would know. Okay, let us go a little bit to your damage hypothesis, that we are producing damage and we can also reduce the damage, which is of course very nice. Can you give the audience a little bit of examples what damage producers are and damage reducers?
Right. So that's also a very interesting question. So perhaps an example is, so let's say you're a smoker at the age of 20, and you stop at the age of 30. So you smoke for 10 years, and that damaged your lung. And so a lot of your cells got turned to these senescent cells, because in our model, our model is actually first discovered on senescent cells in mice. And what happens when senescent cells are increased at a young age is that we can quickly remove them. But what happens at old age is that the mechanism to remove the senescent cells are saturated. So if we let's say if we smoke at the age of if we start at the age of 60 and then stop at 70, the effect of that smoking, 10 years of smoking, will persist much longer. So in fact, we have an experimental version of that where we give a cell damaging agent to the lungs of mice at young versus at old age, and it takes about 5 times longer in the old mice to recover from that.
So that means the term intrinsic capacity. So the capacity is much better at younger age, not so much at older age. So we can do stupid things at young age, but not at old age.
Right, exactly. So I think we have mechanism when we're young to recover from all sorts of things, and that suppresses the variability among individuals. But at old age, because we lose those capacities, so now the differences between individuals become magnified.
Yeah. Okay, so the listeners at a little bit older age, be careful with your body. We always say, okay, there is noise. Can you tell us a little bit what your definition of noise in the system is, and how that maybe even would interact with damage production and reduction? Because very often we treat that as separate things.
Right. So that's also a great question. So I want to mention that, for example, our model holds true in clonal populations of model organisms, let's say simple bacteria, where every cell is of the same genotype. And we also did very careful microfluidic-based experiment to keep the environment constant. And yet this model still applies, and there is still a level of noise and differences among individuals. So this noise should consider to be intrinsic physiological fluctuations. So one example I can give is, for example, stress. So in the normal life, we encounter all kinds of stresses, and these stresses are not necessarily pathological. For example, you have an exam coming up, or you have a date coming up, and suddenly your stress level goes up, and your cortisol starts secreting, and maybe you're not sleeping so well. And this will, in our case, these stresses will go away because the circumstances have changed or that your body becomes adapted to it. So these are what we consider stress noises. But the truth is that we are still quite vague on what this kind of noise is. And I think we actually have some interesting study from Peter Fedoshev that's perhaps can tell us what these noises are.
Yeah, so I think if I summarize, noise can turn into damage producers.
Yes, exactly. So noise is a double-edged sword. So it can lead to high damage, and that feeds in on itself. But it could even have beneficial impacts because at very old age, when everybody is old, you do have occasionally individuals that does much better than their age would predict. And we think this is perhaps related to physiological fluctuation as well.
Yeah. What we do in survival curves is very often dichotomizing if somebody is alive or not. Yes, so it's two groups. We are
Yes.
doing the same, I would say, in our conversations regarding to our health, like it's health span and it's lifespan. Like you are healthy or you are not healthy. We are dichotomizing again. Isn't it that we should taking it a little bit more seriously by making a little bit more categories? For example, taking biological age into account to really understand how our body is aging and when the biological age is increasing over time, and not when it hits a certain mark and then saying, okay, you are dead or alive, or you are healthy or you are not healthy.
Mm-hmm. Yeah, that's a very interesting comment. So our hope is that we can actually develop a sort of a biomarker for not only chronological age, but biological age. And so this will tell you whether you actually need intervention because the situation is quite asymmetrical. So if you are young and almost all of the cases your biological age is going to be small, it doesn't really matter. But if you are middle-aged or more advanced ages, you really have to pay attention to your biological age because like we previously discussed, if you do something stupid, it's gonna hard to recover from that.
Yeah, but would you imagine that the outcome is not dichotomous in the end, but could be depicted as a biological age or a change as a biological age?
That's right. So because the process is exponential, so it's not completely dichotomized. We are describing a biological age with a continuous variable, but this variable is also not linear. So there is some nonlinear effect.
Yeah. Because this is what we are searching for in clinical practice. Patients, clients are asking, okay, how can I modify my biological age, my curve? How can I steepen it or
Right.
not? Depends on what you want to do.
Right.
You showed wonderful data of the NIA ITP program of interventions.
Yes.
We discussed it, or you discussed it based on we are giving an intervention, or we are not giving intervention. Does it matter when you start the intervention in these mice?
That's a great question. It does matter when you start with the intervention, and this kind of experiment would actually, or we consider this kind of experiment would be super informative, where we're shifting from a non-intervened group to a intervened group, and this will give us much more information. But in the present study, because although we do have such experiment in our data set, but for example, we usually have one or two groups. So that's not enough for us to do the study. So we decided to not pay too much attention to it for the moment. But for sure, additional step would be to analyze the dosage dependence and also the regime dependence in the NIA-ITP data.
Yeah, but wouldn't you then, if the hypothesis is right, that at older age we have a steeper decline of function, etc., towards a certain of when we are exposed to a certain exposure, that you could start earlier with later with an intervention when you actually have that more exposure. For example, you start smoking when you're at the age of 60 or 70.
Right. So that's actually a great point. So scaling interventions, so we think that for scaling interventions, you do have to start early to get the maximum effect. But for steepening interventions, we think that you could act because the time scale is relatively fast with damage removal. So you could apply, for example, when you see that you already have advanced biological age, and you can apply this steepening interventions or just remove the bad lifestyles, and then the biological age should fall to sort of the steady state level given by your chronological age. So that's one thing. And the other thing is that we do see some experiment where if you do something bad at old age, sometimes the data gives you some overshoot. So that's also pretty bad.
Yeah. So does it mean that then the steepening interventions are more likely to fit into our conventional healthcare? Like we first see that our system is breaking down, then we are going to the emergency department, then we get the help.
Yes, exactly. So the steepening interventions, what we see is that it correlate with conventional ideas of treating age-related phenotypes like inflammation. So this is clearly what we see in our analysis. And the scaling interventions, while super fascinating, like rapamycin, that the result is super robust. So the steepening interventions, I think it depends on whether you're suffering from a particular problem, for example. So, for example, if you have hypertension, perhaps as the male mice in our experiment does, then the treatment for sure works. But the rapamycin result is very robust. So that's also a very intriguing aspect to us. Why is the rapamycin so robust? And whether we can sort of intervene on that scale the same way we intervene on the steepening interventions. So that's also kind of a future direction.
Okay, so that's a good keyword because you touched on rapamycin, and Dr. Ayla Hodgkins-Courage actually is preparing our longer study. So I will give her the word. But I have one final question left. Look, we do lots of randomized controlled trials. We are measuring the biological age. We do the interventions in humans.
Mhm.
How could you actually kick in to model our data?
Well, so you are getting into what I am doing right now is to model intervention data in humans. And because in model organisms, the most robust data is death. But in humans, we have actually tons of medical data. And our theory actually still applies. So instead of modeling distribution of lifespan, now we have to model distribution of performances. And it's really exciting that we recently discovered that our model can apply to performance data. For example, physiological measurements such as strength or metabolic rate or kidney filtration rate. So our current idea is that perhaps we can use performance data to tell us what the medical interventions like pre-existing drugs, what are their effects on aging, and whether some of them might be steepening interventions.
Okay, so you know where to find us. Over to you, Ayla. I think you are delighted to speak about maybe rapamycin.
Thank you very much, Professor Andrea, for the introduction, and thank you for the lovely presentation today. It sparked many questions in my head at the moment. As Professor Andrea mentioned, I'm mainly working on clinical trials, and I have a question myself. I would really like to learn and see how could your findings be translated into to clinical trial design. So I will go back to the biological age, and I would like to ask you how feasible is it to use steepness as a metric derived from biological aging trajectories in clinical trials as one of the endpoints.
So So indeed, so what steepness tells us in the case of lifespan is that the steeper the curve is, it means every, for example, mice died at the same time. So what we consider to be a steepening intervention has to satisfy two conditions. One is that it's indeed a longevity intervention that improves health and lifespan in a very conventional sense. But the second condition is that the outcome it gives to the individuals are homogeneous, meaning that everybody gets roughly similar amount of improvements. So indeed, this is something that could be done in clinical trials because you usually enroll a lot of individuals, and whatever the endpoint, not only can measure the average, but also the distribution of the endpoint measurements. So in fact, that would be something that's very interesting for us as well.
Thank you. I'm looking forward to maybe working with you on that. And so maybe my second question is, how do individuals' heterogeneity, like genetic background, affect your model's prediction?
Right.
Yeah.
Right. So that's also a great question because this could complicate our results because for now our model only consider genetically homogeneous. So we're not considering the effect of genetic variations. So so most of the variations in our model are coming from physiological fluctuations. So we have to be really careful in terms of that, to ruling out potential confounders on the genetics. But I think what's also comforting perhaps to us is that the aging process, I think, is relatively homogeneous genetically because as far as we know, there is no genes or interventions currently changing the slope of the Gompertz law. So that's sort of maybe not fortunate for research overall, but perhaps it's not going to complicate our study too much.
Mm-hmm. And last one, just for clinical trials, can this model be adapted to N of 1 or personalized aging interventions, where the damage trajectory is actually different for each individual based on their biomarker data?
Yeah, absolutely. So we think that based on both differences in chronological age and biological age, individuals should follow very different trajectories. For example, someone at much worse health and much older ages should respond quite a bit slower to what we consider steepening interventions. So this will be a strong prediction of the model.
Okay, thank you very much. I have some questions from the audience. One of it is, do you think in the future we'll all get treatments to compress Sickspan, like now we take vaccines?
Right. So I think my answer to this is going to be relatively conventional, similar to what's consensus. So you got enroll populations that are healthy otherwise and relatively heterogeneous, so they can't be from the same family and related genetically. And you have to look at a broad range of physiological measurements related to many different systems and not just one. For example, not just to cognitive health, but also physical health and immune health, for example.
Thank you. We have one more question. How can you distinguish between adverse noise and a beneficial hormetic response, particularly when both arise from acute stressors? And how might each differently affect your models?
So that's also an interesting question. So for now, we don't actually have the ability to differentiate different type of noises. If some of you are modelers of stochastic systems, you know that noise can have different characteristics. And for now, we are using mostly standard Brownian noise. So they are symmetric in both sides. So it's definitely a research question for the future.
Thank you. If we wanted to run a 5-year human trial, aim at Sickspan compression, which intermediate biomarkers or digital phenotype would you adopt as an early go, no-go signs before waiting for morbidity data to mature?
Right. So that's very interesting. So I would first I would guess that the endpoint outcome is not going to I'm not going to guess correctly the timescale of the endpoint outcome. So I would try to set my endpoint to differing intervals, for example, in a few months and in a few seasons and in a few years and so on. And then also I would specifically look at inflammaging because that's strongly correlated with many of the biological age predictors. And this is also what we think in humans that's driving a lot of the age-related phenotypes. So I would look at markers of inflammaging.
Thank you very much. We just got a comment, and this was an excellent presentation. There's somebody who's chronologically 53 years old and asking what people of my age should be doing to improve their longevity and health span based on the models you have presented.
Okay. So I'm not so far from this questionnaire in age. So I'm doing all the stuff that people are prescribing to me, to people of my age. So sleep well, don't get so stressed, don't smoke, lose some. I don't know the gender or the sex of the questioner, but basically don't get normal weight, and also exercise. We actually have some exercise data that we analyzed and we didn't put in the paper that clearly show that exercise is a steepening intervention. So this is a it's a very from a very high quality study. So I trust that data.
So which kind of exercise was it, and what was the duration or intensity?
I think it's just for mice, and it's actually aerobic exercise.
Okay. So we have another question. How can we tell if a treatment is actually helping people stay healthier for longer?
Right. So that's quite interesting. So what we're considering to be interventions now are actually our continuous interventions. So it's something you have to do lifelong. We haven't really considered something that's kind of transient. It's some interventions that are transient. So that's also an interesting question. But just speculating here, I would guess that transient scaling interventions actually, such as rapamycin, might have a lasting effect, but that's just speculation on my part.
Thank you. And I think last question, how can your framework for compressing Sickspan in a model organism be adapted and validated for more complex and variable measures of morbidity in humans?
Can you read that question again? I
Yes.
didn't get.
How can your framework for compressing Sickspan in model organisms be adapted and validated for the more complex and variable measures of morbidity in humans?
Right. Okay. Thank you. So this question actually tails very well with what Andrea asked me about clinical trials, because I think in humans we actually have an opportunity where the physiological responses to interventions are much better documented, and also the statistical power of clinical trials is much better than mouse studies. So I think it's actually a great opportunity to apply our model to clinical trials. But I also want to caution here is that the human interventions, what we see is that the maximum lifespan of humans is very robust. So we haven't seen any evidence where there are interventions that can extend the maximum interventions. So perhaps we can find some steepening interventions that's consistent with existing medical advices. But I'm not, I don't expect something that can prolong the maximum lifespan of humans to, let's say, 200 years.
Thank you so much, Yifan. Thank you so much, Ayla, for this very interesting conversation. I would love to hear how you will model the exposome inside of it, because Ayla touched on the NS1 approach. We might all need different interventions, and we are living in different exposures, and how to actually model that, because the mice are treated quite in a homogeneous condition, of course. Thank you so much, Yifan. Big
Thank
applause.
you. This is very pleasant.
Thank you for joining today. As always, there is a chat function. Please use the panelists and all attendees ones to leave your comments, your feedback, and everything you want to share with us. Also, if you have certain speakers you want to see in our show, so please be in touch with us. We have 2 places left for our Healthy Longevity Talent Incubator 2025. It occurs in roughly 2.5 weeks' time. We are looking forward to be in a bootcamp with youngsters all around the world, to actually really discovering not only how we can engage with the healthy aging process, but antagonize aging processes. We will also focus on leadership, etc. If you want to have more information, please have a look at longevityacademy.sg, and there's also a QR code to scan. The next Healthy Longevity webinar is on the 12th of June. And Simland will talk really about a very interesting topic about implementing longevity science into everyday practice. I will absolutely be there. Before closing this webinar with a final video, I wish you all the best, and hopefully not ageism. Take care.
What is ageism?
I've heard of it, but I don't really know what it is.
Ageism? Is that really a thing?
Ageism against older people is stereotyping, discrimination and mistreatment based solely on a person's age. It affects people in many ways and is so common and accepted, most of the time we don't even notice it. But maybe we would notice if it wasn't there. In an Australia without ageism, older people wouldn't automatically be less valued than younger people. With the same qualifications, it won't take them longer to find work, and they won't feel the need to hide their age from potential employers. We will get and not get jobs based on our abilities, rather than outdated stereotypes of what people our age can and can't do. Knowledge will be shared, younger to older, older to younger, for the benefit of everyone, including employers. And older workers won't be forced out because of their age. Without ageism, older people's voices will be heard and given the same respect as everyone else's. Getting older will feel like an achievement, not a failure to be hidden or fixed. Birthday cards with belittling ageist jokes will disappear. Derogatory terms for older people will be as unacceptable as derogatory terms for race and gender, even well-meaning ones that unintentionally patronise. We'll realise only some older people live in nursing homes, only some are hard of hearing, and only some have dementia. Thinking all older people are the same will be as absurd as thinking all younger people are the same. Getting older will be a normal, natural part of life that happens to all of us if we're lucky. Being older can be 1/3 of our life, and one of the best parts of it. Without ageism, we'll live in an Australia that respects everyone and everyone's future self. A place where we can all participate at every age. Older people who are pains in the bum will be treated the same way as younger people who are pains in the bum. No one's asking for special treatment, just equal treatment. Can Australia really get rid of ageism? Yes, we can. All these organisations have already committed to it. Visit everyagecounts.org.au to find out more and pledge your support. And let's make ageism in Australia a thing of the past.
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now. Nothing gonna bring me down.